Hi everyone, welcome to Module 1, in which we are going to discuss the drug targets and relevant drug design issues. This particular video is the first mini lecture out of 4 foreseen in this module, and it deals with the main classes of drug targets. So let's start. First of all, what is the meaning of the term target? Targets are those proteins or other biomolecules such as DNA, RNA, heparin, peptides and so on, to which the drug directly binds and which are responsible for the therapeutic efficacy of the drug. If the drug also binds to or metabolized by another type of biomolecule, which are not known to be responsible for its therapeutic effect, such biomolecules are not defined as targets. The first attempt to compile a definitive target list dates from 1996, when Drews and Reiser, for the first time, estimated the number of human molecular targets for approved small molecule drugs. Starting from that paper, the concept of privileged protein families, with a consistent and successful history of drug discovery, began to emerge. The most recent list of FDA-approved drug targets was compiled by Santos et al. In 2016. They identified 667 unique human protein efficacy targets and 189 pathogen protein efficacy targets. Rhodopsin-like G-protein coupled receptors, or GPCRs, ion channels, protein kinases, and nuclear hormone receptors are considered to be privileged families, given that they alone account for 44% of all human protein targets. These privileged families are responsible for the therapeutic effect of 70% of small molecule drugs. The remaining human protein efficacy targets are mostly unrelated enzymes. In the case of biologics, secreted or surface antigen proteins are the most important target class. Take a look at the figure. The left diagram shows the distribution of human drug targets by gene family, while the right one depicts the distribution by the fraction of drugs targeting those families. As one can easily see, the historical dominance of 4 families, namely GPCRs, ion channels, kinases, and nuclear receptors, is clear. From the lower diagram, we can estimate the clinical success of privileged protein family classes. The distribution of non-approved compounds in Campbell database per family class is shown in purple, while the distribution of approved drugs per human protein family class is demonstrated in orange. This bar chart shows innovation patterns in privileged protein classes. A higher number of recently approved drugs that modulate kinases compared with the number of recently approved drugs that act through either nuclear receptors or ion channels. The total number of approved drugs is shown on top of each bar in bold font together with the number and percentage of drugs approved since 2011, concerning the total number of drugs modulating these 4 families. The important problem of modern drug design is the low rate of new drug target discovery. Currently, the market addresses only a small number of drug targets. Hence, pharma companies have much available pool to exploit. On average, the industry evaluates only about 3 new druggable targets per year. As it has been estimated that only several hundreds of the thousands of potential targets will be druggable and disease-modifying, the pharmaceutical industry is faced with a major challenge to rapidly and reliably identify the most promising targets for drug discovery efforts. If we take a look at the table, we will see that besides human proteins, a lot of FDA-approved drugs also target proteins of pathogens. Most of the FDA-approved drugs are small organic molecules, but in the case of human protein targets, many biologics also serve as drugs. This is not the case for pathogen protein targets, where almost all approved drugs belong to small organic molecules. One more thing that comes from the table is the number of drugs per target and the number of targets per drug. By simple averaging, we obtain the figure of 2 drugs per target. Some targets have provided a ridge ground for selective drugs, such as the glucocorticoid receptors, which has 61 approved drugs. In contrast, others fall into the opposite category, such as kinase inhibitors, for which few drugs act on many targets, thus contributing to the overall pharmacological response to those drugs. Another key developing trend is monoclonal antibody therapies, which are typically highly specific to a single gene product. This contrasts with small molecule drugs for which the interaction with multiple targets is more common. The number of small molecule and biologic drugs per therapeutic area are shown in the table on the screen. As can be seen from the screen, most of the FDA-approved small organic drugs are used to treat cardiovascular diseases and also disorders of nervous system and infections. Most of the approved biologics, in turn, are developed to cure cancer and immunological disorders. Reasonable target selection is an efficient strategy to mitigate risk in preclinical drug discovery. A study of the phase 3 trials showed that 2/3 of failed phase 3 trials between 2007 and 2010 were ascribed to low efficacy. Low drug efficacy is largely due to insufficient target validation at the preclinical stage. The identification of novel promising drug targets with potentially high clinical efficacy is a critical step in the modern drug discovery pipeline, but still remains a great challenge. From a chemical perspective, drugs generate therapeutic effects through altering the expression or activity of target genes or proteins related to the diseases. A substantial proportion of drug discovery efforts in the past decade have involved the rational selection of mechanistic cancer drivers to be targeted. Cancer drivers are proteins that give cells a growth advantage when they are mutated, helping tumors to proliferate. Moreover, cancer is the area of the biggest growth in large-scale systematic efforts to identify disease drivers, powered by major international consortia. So it is interesting to consider the impact of such efforts on the identification of novel clinically validated targets. The 154 cancer drugs approved by the FDA can be broadly divided into 3 groups. 26 drugs are cytotoxic agents, 38 drugs are broadly cytotoxic and act at least partially through protein targets, such as proteasome inhibitors, and 85 drugs can be assigned to clear mechanistic protein targets. Moreover, 5 more drugs act through unknown or non-protein targets. If we compare the lists of consensus 553 cancer drivers to the list of 109 protein targets of the 85 protein-targeted cancer drugs shown in the figure, we can find only a small overlap between the two sets, which is equal to around 30 proteins. Currently available data show that successfully targeted genes share certain evolutionary and genetic features. A successful target means that the corresponding drug of this target has passed the clinical test and successfully marketed. Many human diseases have a genetic component, so genetics can help in differentiating between the causes and consequences of human diseases, which is useful for identifying therapeutic targets. From this simple scheme, one can see the correspondences between genetically caused changes in target function, which can be a gain of function or loss of function, and drug mode of action, such as stimulation or inhibition, that lead to therapeutic and side effects. Kwan et al. In the paper published in 2018 revealed that drug target genes did not arise smoothly during macroevolution, but are enriched in genes that originate in prokaryotes, metazoa, and eumetazoa. Successful drug targets are concentrated in genes that emerged at certain evolutionary stages. This feature can prioritize human genes during the selection of drug targets. Moreover, the gene age of drug targets also could be the signpost pointing to certain diseases. Several diseases, such as cancer and neurological disorders, are associated with genes that originated in certain evolutionary stages, providing abundant source of the corresponding drug targets. A combination of the evolutionary and genetic features of drug targets can better facilitate target identification. An ideal targeted gene should contain a series of functional alleles that are related to function and clinical outcome. Essential genes are a subset of genes in the genome that have central functional roles and are required for the survival of an organism. As such, they are involved in crucial developmental, metabolic, and signaling pathways, tend to be located more centrally in protein-protein interaction networks, are more highly expressed, and are more conserved throughout evolution. However, less is known about the essentiality of drug targets. It has been suggested that drugs targeting non-essential proteins have higher approval rates, probably because these proteins are safer to target. There are two principal groups of drug targets: genetic targets and mechanistic targets. The genetic target focuses on the genomic content of the pathogen or cell, modulation of which alters the genetic program. It should fulfill the two conditions. First, the disease caused by genetic mutations or increased disease risk attributed to a genetic factor. Second, it must contribute to disease prognosis. The use of genetic targets for drug discovery has been limited, as most of the diseases are multifactorial. Thus, the nucleic acid can be an appropriate target, but because of its complexity, nucleic acid targeted drug design has not been a major path opted in pharmaceutical discovery programs. The mechanistic targets represent biomolecule inhibition of which was sufficient to obtain a significant therapeutic effect. The drug targets were identified based on biological observations and clinical findings. For example, receptor or enzyme altered in a disease condition. It also includes the membrane-associated proteins, kinases identified in disease models. Membrane proteins, such as receptors and ion channels, are key molecules in the membrane that account for up to 2/3 of non-druggable targets. Thus, they represent an important domain for pharma. For example, GPCR accounts for over 50% of all human drug targets in cancer, CNS disorders, inflammatory diseases, or cardiovascular diseases. Now we will briefly discuss the structural features of the most important protein drug classes. First of all, let us consider the G-protein coupled receptors. GPCRs belong to a large family of signaling proteins that mediate cellular responses to most hormones, metabolites, cytokines, and neurotransmitters, and therefore serve as fruitful targets for drug discovery. GPCRs are categorized into 6 classes based on sequence and function, namely Class A, which comprise rhodopsin-like receptors, Class B, the secretin family, Class C, which includes metabotropic glutamate receptors, Class D, which comprises fungal mating pheromone receptors, Class C includes cAMP receptors, and Class F comprises frizzled and smoothened receptors. All GPCR members share a common 7 transmembrane architecture linked by 3 extracellular and 3 intracellular loops. Different classes of GPCRs have low sequence identity and possess different extracellular and terminal domains and diverse ligand binding pockets. In the case of class A GPCRs, the endogenous ligand is recognized by a ligand binding site in the 7 helical transmembrane region. For class B, the ligand is recognized by both extracellular loop and 7 helical transmembrane domain. For class C, the ligand binding pocket is found in the extracellular domain that contains a venous flytrap module. In the case of class F GPCRs, both smoothened and frizzled receptors possess an extracellular domain that is comprised of an extracellular cysteine-rich domain and an extracellular domain linker domain. The endogenous lipoglycoprotein ligand VNT binds to the CRD of the frizzled receptors. Upon ligand binding, GPCRs activate at least one of the 2 signaling partners, namely heterotrimeric GTP binding proteins or G proteins or beta-arrestins, and mediate signal flow via modulation of various downstream effectors. Class A GPCRs are the most immensely investigated GPCR drug target within the drug market due to their centrality in diseases, structural availability, and relative ease of accessibility. The high druggability of GPCRs and its central role in numerous diseases provide a strong spearhead for its continuous efforts in drug discovery and development. A recent study of all GPCR drugs and agents currently in clinical trials revealed that 475 drugs mediate their effects through 108 unique GPCRs. The first structural breakthrough of a human GPCRs, IDEST beta-2 adrenergic receptor, using different crystallization techniques, was solved in the year 2007. Moreover, the first crystal structures for GPCR classes B, C, and F have been also solved. So far, experimental structures of 44 distinct GPCRs and 205 ligand receptor complexes covering all the 4 classes A, B, C, and F are available, of which most belong to the class A subfamily. GPCRs are known to exist or function as monomers, dimers, and/or higher-order oligomers, including homo or heterodimers and homo and heterooligomers. The oligomeric structures of GPCRs are essential for modulation of receptor function, mediation of crosstalk between GPCRs or other signaling pathways, and cellular trafficking. Hence, they have been associated with specific functional effects. Moreover, targeting these oligomeric structures as drug candidates could provide a new arena for drug development and specificity. Now let's talk about kinases. The tertiary structure and overall folding of the kinase catalytic domain are highly conserved throughout the kinase family and harbors around 300 amino acids. The first crystal structure of protein kinase A in complex with ATP and the pseudo-substrate in 1991 paved the way for a better understanding of the catalytic mechanism, cofactor binding, and kinase regulation. The canonical kinase catalytic domain fold consists of 2 domains, also called lobes, which are connected via a flexible hinge region. Both lobes form a hydrophobic cleft, which serves as a binding site for ATP. The smaller N-terminal lobe comprises 5 beta sheets and 1 helix called alpha C, whereas the terminal lobe, C-terminal lobe, is mainly alpha helical in structure and called alpha D, alpha E, alpha F, and alpha I. A hallmark of the active kinase conformation is the accurate spatial arrangement of the conserved catalytic domain motifs for efficient catalysis. In the picture, you can see the architecture of the kinase catalytic domain. At the left is the conserved tertiary structure of a protein kinase exemplified by cyclin-dependent kinase 2 or CDK2. Catalytic active site of CDK2 in complex with ATP and a peptide substrate. The P loop is highlighted in pink, the alpha C helix in blue, the hinge region in yellow, the activation loop in green, ATP in orange, and the peptide substrate in black. The magnesium ion is shown as a green sphere. The N-terminal and C-terminal parts are marked. In the right part, you can find details of the ATP binding site. ATP and substrate are shown in stick representation. Key hydrogen bonds are shown as dotted lines. The ATP cofactor complements the catalytic spine or C-spine with an aromatic interaction of the adenine ring system, bridging both kinase lobes. In inactive kinases, the spines are broken, leaving one or more structural motif disconnected from the active state position. Normally, the N and C lobes adopt a closed conformation with a structured P loop. At this figure, you can see the alignment of hydrophobic spines in the catalytic domain of active protein kinase A. C-spine is colored in gold and R-spine in green. The bridging residue between both spines in the N-terminal lobe is highlighted in pink. Another important protein target family is ion channels. They are pore-forming protein complexes that facilitate the flow of ions across the hydrophobic core of cell membranes. Most sodium, potassium, calcium, and some chlorine channels are gated by voltage, whereas others, such as some potassium and chlorine channels, transient receptor potential channels, ryanodine receptors, and inositol 3-phosphate receptors, are relatively insensitive to voltage and are gated by second messengers, and also other intracellular or extracellular mediators. Many ion channels share several structural similarities, which suggest that they have evolved from a common ancestor. The figure represents the amino acid sequence relations of the minimal pore region of the voltage-gated ion channel superfamily. This global view of the 143 members of the structurally related ion channel genes highlights 7 groups of ion channel families and their membrane topologies. They are shown in different colors and different lobes of the figure. 4 domain channels, like calcium V and sodium V, are shown as blue branches. Selective potassium channels are shown as red branches. Cyclic nucleotide gated channels are shown as magenta branches. And transient receptor potential and related channels are shown as green branches. Background colors separate the ion channel proteins into related groups. Light blue, calcium and sodium voltage-gated channels. Light green, TRP channels. Light red, potassium channels. Light orange, KV, 10, 11, 12 channels, and cyclic nucleotide modulated, cyclic nucleotide gated, and also hyperpolarization activated cyclic nucleotide gated channels. Other ion channels such as chlorine channels, aquaporins and connexins have evolved separately and possess completely different structural properties to the VGL channels. It should be mentioned that ion channel modulators are an extremely successful drug class, second only to drugs targeting GPCRs. To sum up, let us recall some of the key points of this lecture. First, rhodopsin-like GPCRs, ion channels, protein kinases, and nuclear hormone receptors are considered to be privileged families of drug targets. Some targets have provided a rich ground for selective drugs, whereas others fall into the opposite category. The pharmaceutical industry evaluates only around 3 new druggable targets per year. Only a small part of known chemical space is covered by drugs targeting specific proteins. There is a small overlap between the list of known cancer drivers and already exploited protein drug targets. There are predominant groups of diseases which are covered by most of the available drugs. Among them are cardiovascular diseases, systemic infections, nervous system disorders, immune and neoplastic related disorders, and alimentary and metabolism disorders. All GPCR members share a common 7-transmembrane helical architecture linked by 3 extracellular and 3 intracellular loops. The canonical kinase catalytic domain fold consists of 2 domains, also called lobes, which are connected via a flexible hinge region. Both lobes form a hydrophobic cleft, which serves as a binding site for ATP. Most ion channels are gated by voltage, while others are voltage insensitive and are gated by secondary messengers and other mediators. On this slide you can see some suggestions for further readings. So that's it. Thank you for your attention, and now we suggest you to proceed to lecture 1.2 on the assessment of protein druggability.